tissue culture. This one is introduced by Haberland in 1902. He used a Knoop's solution, salt solution. Now we are using MS solution, Murashige and Smith solution. Okay. Some terms: explant, callus, then organogenesis, and one more term is there, and that one is clones and clones. Tissue culture is a new technique. This technique is what we are calling is another technique. That one is helping to keep pace with the increasing food demand of the people. We must note here, this activity was first introduced by this Haberland in 1902. In this case, in this activity, what he did, he used, in this case, in tissue culture, isolated protoplast, cell, tissue, or the plant part is utilized for generation of a new plant. Why this type of thing were considered for generating the plants? In earlier case, we paid attention on plant breeding. That one is a time-taking job. But this is in very short span of period, we can generate multiple of crop plants. In this case, we must note, here plant cell has a potentiality to generate a single single cell can have a potentiality to generate an entire plant. This is totipotency. This ability is considered, and by considering this ability, this branch flourished. Before entering in tissue culture, its steps and the procedure, some terms are being used in this tissue culture. These terms are, the term one is explant, another one is callus, third one is organogenesis, and the fourth one is clones. Explant is what? This is excised part of the plant. It may be from root, it may be from stem, it may be from tubers, or it may be from the meristematic tissue also. Generally, the permanent cells are taken, permanent parenchymatous cells are taken. Permanent, the meaning is they lose the ability, they lost the ability to divide and redivide. But that ability regains later on, and new plant generation could take place. New entire plant generation could take place. Here, explant we have just now mentioned, this is excised plant part, no more than this one. The plant we are here expecting to multiply, that plant be acting as a donor plant, and the plant parts from that donor plant we have taken is an explant. This may be a protoplast, this may be a cell, this may be a tissue, small part, or the organ of the plant. be utilized here. This is explant. Callus is nothing, this is the unorganized mass of cells. Here all cells are morphologically similar, unorganized one, no division of labor is found out in this group of mass of cell to whom we are calling the callus. Organogenesis is another term where the different parts of the plants are developed. Here is generation of root, generation of stem. Generation of root is called as rhizogenesis and generation of stem is called as caulogenesis. Root generation and stem generation takes place to the callus is organogenesis. And the clones are nothing but these are the carbon copies of the what we are saying the donor plant. Multiple copies having same genetic makeup, because these are calling clones because all the cells multiplying are here by the act of mitosis. With the help of mitotic division or the somatic cell division, no exchange of genetic makeup takes place, and that's why the generated all members, they have similar genetic material. We are calling them as clones, the carbon copies. And this is what the different terms may come while studying this tissue culture. Here, what are the requirements needed for tissue culture technique? Requirements for the tissue culture. What things are needed? And thing one is the donor plant, then the medium is needed. That may be solid or liquid. Third one is, with this one, the minerals as well as the what we are saying here. Medium is needed, and the laboratory conditions as well as facility, aseptic laboratory conditions. Then the one more is needed here, and that one is facility to control different factors and the growth factors. Facility to control different physical factors, like temperature and the growth factors. Here, such different requirements are needed for the tissue culture technique. Donor plant provides here explant medium on which this explant is being implanted or kept, where that explant receives, where from the explant receives nutrient as well as growth factors, and the aseptic laboratory conditions are also necessary here. Without this aseptic laboratory conditions, the tissue culture could not be possible. Either that may contaminate the tissue, that may contaminate the, or may totally vanish the activity. Here facility to control different factors like temperature. Here one can say the light, humidity. All these factors are needed here, and the last one can add here, and that one is various growth factors, especially two growth factors like growth hormones are utilized. One is auxin and another one is cytokinin. Here, these are the requirements. Nutrient or culture medium being used. In the medium used, culture medium, I have mentioned here medium, precisely culture medium. We know now the donor plant, they produces, here they provides, they expand. The culture medium is needed. How this culture medium is? Nutrient medium is. This nutrient medium must be available. This nutrient medium, culture medium is available in the market with various concentrations of the inorganic and organic molecules. Here that one is either liquid or the solid also. Besides this one, that medium may contains sugar molecule, may contains here the sugar molecule or sucrose we may call, and the growth hormones here. Haberland, the father of tissue culture, he used a Knoop's salt solution. Now we are using MS solution. This is Morashige and Skoog solution. Here that solution, which one is liquid, may get solidified here by adding a polysaccharide from red algae. This is called as agar-agar. Agar can be utilized and the medium B solidifies. Solidification or semi-solidification is taken out, is carried out as per our need, and here medium is kept ready for the tissue culture activity. Sterilization is one more activity. If the sterilization is not proper, the contamination may take place and the entire experiment may fail. Here for this one, sterilization may take place. Sterilization of what? Sterilization of various utensils we are using in this experiment. Here's all should get sterilized here. Various the utensils as well as the medium and the explant all should get sterilized. Normally the explant is sterilized by 1% sodium hypochlorite, or one may say here the 70% ethyl alcohol can also be utilized, or 10% hydrogen peroxide can also be utilized for what purpose? For sterilization purpose of the explant here. With this one, here the sterilization technique of the other utensils is, it may be wet and dry process. It may be either wet process or dry process. Sometimes here alcohol sterilization is also carried out. Here what is the methodology, how the tissue culture is brought into being, how the tissue culture experiment is carried out. What the methodology is? Methodology is very simple. Take explant, allow this explant to generate its own colors. The generated colors forms organogenesis, and later on here new plantlets gets formed here. The explant is, firstly, in methodology explant culture takes place. In explant culture, the isolated protoplast can be utilized. Cell, single cell can be utilized. Tissue can be utilized, or the plant organ is also being utilized. This is what? Different types of explant are taken out and that can be utilized here. And this one is before utilization properly sterilized here. Proper sterilization when takes place, then only this could be utilized. Here not only the explant be properly here the sterilized, and here the medium is also gets properly sterilized and kept, the explant is kept on the nutrient medium, culture medium. Nutrient medium, culture medium that already gets sterilized properly. Then what happens? The content present in the culture medium or nutrient medium, we have mentioned earlierly, the various proportions of the inorganic and organic salts are there, sucrose is there, and the growth factors are there. Cells, the tissue or the plant part, which one is kept on the medium, sterilized medium, that tissue draws nutrition from the medium and it starts growing. The growth, in that growth, cell division takes place, cell division, mitotic cell division takes place, and what happens? All the cells are vegetative cells, forms a mass of unorganized, without differentiated, unorganized mass of cells gets formed, to whom we are calling callus. This callus is unidentified cells are there. All cells are similar, parenchymatous, no doubt. Now, once the callus formation takes place, here in the medium, changes takes place. Changes in the growth medium could take place. The changes in the growth medium, here growth medium like two growth hormones are there, two growth hormone. In case of plant tissue culture, auxin and cytokinins are utilized, used here. Auxin, when exceeding the concentration, then the cytokinin, here the root formation takes place. When cytokinin is exceeding, then the auxin, then the kaulogenesis, stem generation takes place. That type of rhizogenesis and kaulogenesis could bring into being by the change in proportion of what the hormones of the plant here. After the development of the organs, here the plantlet generated and that plantlet later hardens in the laboratory and here allowed to plant in the field. But a plant is giving us the explant and its multiple copies when we are in a mood to generate. We could develop a suspension culture. Suspension culture, for formation of suspension culture, liquid nutrient medium is needed. In liquid nutrient medium, no solidifying material agar-agar, the polysaccharide, is used here. And the callus is kept in the nutrient medium, suspension medium, and that one is agitated 100 to 250 revolutions per minute. Because of continuous agitation, what happens? The medium gets mixed properly, one thing. Second thing, because of mixing, all the contents are properly nourished. Nourishment, proper nourishment may occur here. With this one, aeration also takes place. Aeration is needed for the living cells which undergoes aerobic respiration. And the third thing is that here no cell could get attached with each other. Cell could get separated. Separation of cells could be possible. Aeration is proper one, nutrition is proper one, in suspension culture could take place. And what happens? Here the number of cells are found suspended in the culture medium, in the suspension medium. Such a medium is fast growing one. And that's why here that one is changed it's faster medium, and it's in that medium is changed normally. This is what we are calling the suspension medium, suspension culture. Here in the cell suspension culture, following advantages are there. We have mentioned that advantages. Cells can be cultured on larger scale. Suspension culture is more or less homogeneous, and the culture do not show the differentiation. It is easier to use for subculturing by the callus and suspension culture. We can achieve cell biomass production, biochemical isolation, regeneration of new plantlets, and formation of transgenic plants and protoplast culture. This is what the tissue culture technique is.